picking up where we left off last week. The last thing we were talking about was the way that we carry oxygen in our blood, okay, which we learned that there were two ways. Dissolved, which was only about 1%, 1.5%, something like that, and then that left us with 98.5% or so bound to hemoglobin, what we call oxyhemoglobin. And I liked the analogy of a piggyback ride to describe the relationship between hemoglobin and oxygen, okay, where hemoglobin is providing a piggyback ride for oxygen from our lungs, where we pick up the oxygen, to our tissues, where we unload it. Okay. Now, we learned just at the very end of the lecture, the word affinity described how tightly oxygen held on to hemoglobin. Okay. And this relationship of oxygen and hemoglobin was described with this oxygen-hemoglobin dissociation curve. And we learned that this curve can shift to the right and to the left. Okay. If we shift it to the right, it's a result of some things. Okay. But that lowers affinity of oxygen to hemoglobin, meaning that oxygen holds on more loosely. Okay. Which means that we load less in the lungs, but we unload more to the tissues. Okay. And the opposite for a shift to the left, where we increase affinity or affinity goes up, causes more loading but less unloading. Okay. So now we get to talk about the Bohr effect and see what things can actually shift the curve. Okay. Our Bohr effect describes the effect of blood pH on dissociation of oxyhemoglobin. Okay. If we decrease pH, it's promoting dissociation. Okay, because pH of 7.2 is low compared to 7.4, as we see here in our figure, causes a shift to the right. And the shift to the right can occur like what happens when we exercise, when we produce lactic acid. Okay. And if lactic acid builds up in our blood, pH will decrease because of the increase in hydrogen ion. Okay. The affinity of hemoglobin for O2 decreases, meaning that we load less in the lungs, because if we draw a line over here from about 100 millimeters of mercury, okay, we're only loading about 80%. We're having 80% saturation. But if we draw a line over from about 30 millimeters of mercury, our dotted line shows that we unloaded more than we did here at normal. About 50% normal, under 40% for a shift to the right. A shift to the left does the opposite. We load more, we unload less. Okay. But a drop in pH, which like happens when we exercise, will shift it to the right. Because it makes sense, hopefully, we want to unload more oxygen to our tissues when we exercise. Because our working tissues, our skeletal muscle, needs more oxygen. Okay. When we exercise, we also produce more CO2. And so CO2 levels will go up. We describe CO2 levels with PCO2, partial pressure of carbon dioxide. And as you see here at the bottom, partial pressure of carbon dioxide goes up, or a shift as a result of high PCO2 is to the right. And that does the same thing, means less loading, more unloading. Okay. Whereas a lower PCO2 shifts it to the left, more loading, less unloading. What happens to your temperature in the body as you exercise? It goes up. Guess what an increase in body temperature does to the curve? It also shifts it to the right. Okay. Why does our body temperature go up? Well, heat is a byproduct of all metabolic reactions in cells, and heat is produced by active muscle cells, especially when we're exercising, where those muscle cells are really active. During hypothermia, where body temperature is low, okay, the shift occurs to the left. You can see that's very drastic here. Okay. We don't need as much oxygen because metabolism slows down. Okay. So if you ever wanted to lower your metabolism, you can just decrease your body temperature. Okay. Obviously, you want to be careful. You don't want hypothermia from setting in. Okay. And then the last thing here is BPG, a chemical also known as 2,3-BPG. It's a chemical that's produced by our red blood cells during glycolysis. If it is increased, it will bind to hemoglobin as it builds up, and then that prevents hemoglobin from binding to oxygen, so that decreases affinity. So it's just the same thing as an increase in body temperature, or a decrease in pH, or an increase in CO2. Okay. It means that there's less loading and more unloading. Okay. Now on the test, you'll need to know what a shift to the right means, what a shift to the left means. You'll also want to know what shifts it to the right or to the left. Okay. So I mainly focus on shift to the right, but for all these, if you just reverse it, it's a shift to the left. Okay. So things that shift it to the right: drop in pH, increased PCO2, increased temperature, and increased BPG levels. Shift to the left will be the reverse of all that. That's it for oxygen. Now we'll talk about the way that we transport carbon dioxide. CO2, we remember, is produced in our tissues, so therefore systemic tissue PCO2 is higher than our arterial blood, pushes the CO2 into our blood, and then that blood that arrives in our lungs has a higher CO2 level than our alveoli, pushing it into our lungs and then out into the environment. So we've created a pressure gradient, established this time the opposite direction as we did with oxygen. We carry CO2 in our blood 3 different ways. It's dissolved, just like we see oxygen was. More CO2 can be dissolved in our blood, about 7 to 10%. CO2 can also bind to hemoglobin to form carbaminohemoglobin, only about 20%, which leaves us with about 70%, the majority, which is in the form of bicarbonate ions. Okay. Bicarbonate ions are abbreviated HCO3, and this is formed when we take carbon dioxide and hydrate it, which means that we add water to it. That forms an acid called carbonic acid. Carbonic acid then dissociates to hydrogen ion and bicarbonate ions. And this all occurs because of an enzyme called carbonic anhydrase in our red blood cells. Now, most of that hydrogen ion will bind with hemoglobin, which helps buffer intracellular pH. And we'll see a figure on the next page to demonstrate this. The bicarb will leave the red blood cell and enter the plasma, okay, because there's a concentration gradient for that. Okay. As that bicarbonate diffuses out, there's an electrical gradient that's established that then pulls chloride in to the red blood cell, and so that's why we call it a chloride shift. When we get to our lungs, we do the exact opposite of what we just did. See how these arrows point in both directions? Okay. The bicarbonate will go back into the red blood cells. Chloride will leave. We call that the reverse chloride shift. It combines with hydrogen ions, forms that acid, which then dissociates this time to water and CO2, and then the CO2 can be exhaled. Okay. Now it's important to understand, and this is going to be something that we'll talk about here a little bit more in just a second, but hydrogen ion can be thought of as a product of CO2 dissociation. So if CO2 increases, so does our concentration of hydrogen ion in the body, okay, or in our blood, as we're saying here, causing a drop in pH. Okay. This drop in pH will stimulate the carotid bodies, which are receptors that we're going to talk about more about here in a second, that increase ventilation, so we increase our breathing rate in order to rid more CO2, thus decreasing the amount of hydrogen ion in our body, or in the blood, excuse me. All right, so let's look at oxygen first. Inhaled into our alveoli, brought in from our alveoli into the blood, most of which is carried by hemoglobin in the form of oxyhemoglobin. Okay. And then when we get to our tissues, we do the opposite. We unload it to our tissue cells. CO2 is produced in our tissues. It comes into our blood, across that systemic capillary wall. Some of it's dissolved into the blood. Some of it is brought into the red blood cell and combined with hemoglobin, to form carbaminohemoglobin. The majority of it combines with water, forms the acid, dissociates to bicarb and hydrogen ion. The hydrogen ion binds hemoglobin. Bicarb comes out into the plasma. As that does, chloride comes in. That's why we call it chloride shift. Then when we get to our lungs, we do the opposite. Bicarbonate comes back in, chloride leaves, there's a reverse. The hydrogen ion combines with that bicarbonate, forms the carbonic acid, that same enzyme, then turns the reaction in this direction to form H2O and CO2. The CO2 is then exhaled out as it enters our alveoli, and then we breathe out. Fun stuff, right? All right, now we get to talk about how we control our breathing. Remember, ventilation is a fancy word to describe breathing. Okay, and we maintain the correct concentration of certain blood gases by way of changing our rate and depth of breathing. So how quickly and how deeply we breathe in. Okay, it's all feedback loops, just like we've seen earlier in the semester. So we've got sensors or receptors that monitor chemical concentrations. Control centers, these are up in our brain. Then we've got effectors, which are the respiratory muscles that we talked about the other day. So here's the input, here's the output. The response, as we see with the dotted line, is going to be negative feedback, okay, trying to return us back to normal. So let's talk about those control centers and sensors. Back when we learned about the brain, we learned that our medulla and our pons were two parts of our brain that controlled breathing. Okay, we had the medulla that had the medullary respiratory or rhythmicity center, okay, that controls our basic rhythm of breathing, okay, or just basically helps us breathe the correct amount of times in a minute, if we want to think of it that way. Within our pons, we learned about the pneumotaxic and apneustic areas. Okay, the apneustic areas in the lower pons, this center sends stimulatory impulses to our medullary rhythmicity area or center, activating and prolonging inhalation. This is what will result in a long, deep inspiration. The pneumotaxic in our upper pons helps turn off the inspiratory area before our lungs become too full of air. So when our pneumotaxic area is more active, our breathing rate is more rapid. We also know that our cortex, the very upper level part of our brain, the very most sophisticated part of our brain, controls a lot of things. It can override our medulla and our pons. Okay. So we can voluntarily breathe faster if we want to. We can also voluntarily hold our breath if we want to. Why would we ever want to voluntarily hold our breath? Go underwater, okay, which I do every day when I take a shower. I also have to plug my nose and hold my breath when my dog lays a wonderful dog fart in the house. Okay, so because as it states there, irritating gases we don't want to hit our lungs. Okay, so we can not breathe, but is that forever and ever and ever? No. So our ability to not breathe, however, is limited by the buildup of CO2 and hydrogen ion in the body. Speaking of, those are some of the chemical concentrations our chemoreceptors monitor. We classify the chemoreceptors as either central or peripheral, based on where they're located. Okay, our central chemoreceptors are in our medulla or near our medulla, and are surrounded by brain extracellular fluid, so our cerebrospinal fluid. Respond to changes in ECF, concentration of hydrogen ion. Okay. Increasing hydrogen ion concentration will stimulate ventilation, whereas a decrease inhibits it. Okay. Our peripheral chemoreceptors, which are located pretty much in the same place as the ones that we talked about for our blood and blood pressure, carotid bodies, bifurcation of the common carotid arteries and the aortic bodies above and below the aortic arch, monitor three things of our blood: the pH of our blood, the CO2 or PCO2 of our blood, and the PO2 of our blood. Okay. And they will be activated when our pH drops, when our PCO2 goes up, and when our PO2 goes down. Okay. If I asked you guys all to hold your breath, those three things would all happen. Because think about it. If we're not breathing in, what's going to happen to PO2? It's going to go down, right? And we're not replacing the oxygen that's getting utilized. If we're not breathing out the CO2, what's happening to PCO2? going to go up. And what did we just learn happens when CO2 combines with water? It forms hydrogen ions. And what happens when hydrogen ion levels go up? Your pH goes down. So, as a result of increasing PCO2, decreasing pH, and or decreasing PO2, the central and peripheral chemoreceptors will stimulate the inspiratory center, causing hyperventilation. So what's hyperventilation? Is that more rapid or less rapid breathing? More rapid. That means we'll bring in more O2, we'll pull out more CO2, and that will increase PO2, decrease PCO2, and actually will increase pH. And that means we're demonstrating negative feedback. And that's what we're showing right here in this figure. Other influences. Our proprioceptors that we've talked about several different times in this class. We learned about those back when we talked about like our stretch reflex. Those are receptors located in our joints, our muscles. They also, in this case, can stimulate us to breathe when we begin exercising. Okay. They help us increase the rate and depth of breathing even before we see changes in those three chemical concentrations. Okay. Why? It's because we're trying to get in the head and stop too drastic of changes occurring in PO2, pH, and PCO2. Okay, so as we start exercising, right away we're going to start breathing faster and more deeper. Okay. An inflation reflex. We have stretch-sensitive receptors called baroreceptors in the walls of our bronchi and bronchioles. When they are overstretched during overinflation of the lungs, nerve impulses are sent along our vagus nerves, short inspiratory and apneusic areas in the brain. These brain regions are inhibited, promoting exhalation. This exhalation will reduce the stretch based on those baroreceptors, or placed on those baroreceptors, and the stimulus is stopped. So it's essentially just like the baroreceptor reflex when we looked at blood pressure. When it went up because they were stretched more, they tried to get the stretch down. Okay. The limbic system we know is what part of our brain? What's its nickname? Emotional brain. Okay. So having various emotional or anxiety levels will stimulate the limbic system, which can then send signals that increase our rate and depth of ventilation. Temperature increasing, body temp increases the rate of inspiration, decreasing it decreases the rate of inspiration. A really sudden cold temp drop will take your breath away. That's what apnea is, like jumping in a cold pool literally will take your breath away. Pain, sudden severe pain causes apnea, whereas prolonged pain will increase the respiratory rate. Irritation of the airways will cause immediate cessation of breathing, followed by coughing or sneezing. And blood pressure, sudden elevation of BP will decrease respiration rate, whereas a drop in BP increases it. And I saved the best for last. I don't know how anybody ever figured this out, but stretching the anal sphincter muscle increases the respiratory rate, which in the book they talk about if somebody is not breathing a great way or if their breathing is really shallow, you can activate this reflex the old-fashioned way. Sticking a finger up the anus. They talk about it not so much in adults, but like in babies if there's a severe situation. Don't go home and practice that. All right, the very last thing I want to talk about just briefly is oxygen consumption. We abbreviate as VO2, where the V here stands for volume. This is the amount or volume of oxygen consumed by the tissues of the body. Okay, it should not be any secret, or this should not be surprising that as our body performs more work, like during exercise, the demand for energy increases, thus increasing the amount of oxygen extracted from our blood. Okay, so in simple terms here, aerobic respiration increases, increasing oxygen extraction. VO2 max is our maximal oxygen consumed by the tissues of the body. It is the best measure of our cardiorespiratory fitness. Okay. Predominantly determined by genetics. Okay, we're all products of our parents in this case. It can be improved with training, but I'm guessing we're all sitting in this room because, you know, we're not elite level VO2 max athletes. Otherwise, we'd be probably training for the Olympics or something like that. Now, at rest, which you would be over here on the left side of the graph at 0, because 0 would be intensity, and 0 is rest. Oxygen consumption is not 0 because we have to breathe and consume oxygen even at rest. Not very much, about 1/4 of a liter a minute. Okay, but if we increase intensity, there's a very linear rise in VO2 up to a point where it plateaus off, and that's what VO2 max is. Okay, the higher the point of plateau, the better off we are as far as endurance goes. Okay. And it's, like I said, very linear. Okay. It increases around a rate of 10 milliliters per watt per minute. So if you ever wanted to calculate it, you could. Not that I would ever ask you that. But over on the right, you can see a table showing different VO2 max values with healthy, diseased populations. And sedentary, we can kind of think of as diseased because they are very, very lower compared to others. Any questions on respiration?